Right. And the first talk, our Lyell medal winner today, Rosalind Rickaby, coccoliths coasting through the Elderfield curve.
Right, I'll hold it together this time, I promise. Okay, so what I wanted to present today is a kind of slightly new view for paleoceanography. When people tend to think of paleoceanography, they tend to think of foraminifera. These are the sort of hundreds of micron-sized calcite particles that we can separate out of sediments. And I guess what I want to show is that the coccolithophores, which make up the other carbonate half of the sediments, and have been viewed very much as the poor or the sort of black sheep of the family, really, because they have such a large biological imprint on their geochemistry. What I want to show is that actually that biological imprint might be useful, and these things that get termed vital effects, the deviation of these mineral chemistries away from equilibrium, may be useful and actually tell us about evolving biology in response to changing climate. And so, as you saw, Harry means a lot to me, and I used his favourite curve where he showed our proxy confidence factor, so how well we believe a proxy. We tend to start out very optimistically, we then start ferreting around in the guts of that proxy, it plummets into this pessimism phase, and that's where the coccolithophores have been really since the 1980s. But then they start to get back into a realism phase. And I guess that for the coccolithophores, I think it's very much a way of changing our minds about these organisms and realizing they may not be telling us about equilibrium temperature signals, but we can still learn a huge amount. This started out during my PhD where I was working on foraminifera and the uptake of cadmium into foraminifera. I'm just showing here the experimental partition coefficient for these two trace elements, cadmium and magnesium, into inorganic calcites, where I'm showing these in the black blobs for the cadmium going into an inorganic calcite and the magnesium being taken up into an inorganic calcite, along with temperature. And this has been the basis of the fabulous paleothermometer that Carrie's been pioneering. And I was very much interested in the cadmium, but the curious thing that I discovered was that if I measured the cadmium in planktonic forams from the ocean in the hope of getting at a nutrient signal, because the cadmium looks very much like phosphate, I discovered that the uptake of the cadmium into the calcium carbonate actually increased enormously with temperature. And this was going totally against what we would have predicted from equilibrium calcite, from inorganic calcite. And so this for me was the real slap in the face that these paleoceanographic signals are not being preserved in inorganic particles, they're being preserved by biology. And both the absolute value of these partition coefficients, so you can see the sort of orders of magnitude in the reasonable realms of Earth's surface, there are orders of magnitude difference between the inorganic uptake of cadmium and what we were finding in our biological particles, our foraminifera. And then the sensitivities to these environmental factors are also very different. So we're finding this positive temperature relationship where the inorganic calcite predicts an inverse relationship. And again, for magnesium, we see real differences between this inorganic calcite and the biogenic particles, a very much higher temperature sensitivity, which in many ways is great for us as paleoceanographers. So the real challenge is, well, how can we then start to build a quantitative context to these proxies if we have these great uncertainties? We can do empirical calibrations, but is that good enough? Could there be other biological factors, maybe nutrients, which are playing into changing the way these organisms biomineralize, and hence the signals get taken into the calcite? And this is where the coccolithophores became very interesting, because these are organisms we can grow easily in the lab. They're sort of like yogurt. You take a goop of coccolithophore, put it into some fresh seawater, just with phosphate and nitrate. I still find it amazing that you can just put chemistry in a solution and these things will grow with a bit of light. But that's very much easier than the foraminifera, where you have to go out and pluck a single foram and observe the addition of calcite, and they never reproduce in the laboratory. So this is where we were back in 1980 with the coccolithophores, and we've got these enormous coccolithophores, the Coccolithus pelagicus. This is a very rather ancient form of the coccolithophores, and we've got Emiliani Huxleyi. You can see these big changes in the size of the coccolith, as well as the degree of calcification. So Coccolithus pelagicus here has got very large, crunchy platelets, whereas EHUX is somewhat more fragile in its calcium carbonate. And here we've got the dependence of the oxygen isotopes in this calcium carbonate with temperature as predicted by equilibrium for inorganic calcites. And then for these small, lightly calcified groups, we see that they were termed the isotopically heavy group. So they were offset by 1 or 2 per mil from this equilibrium paleotemperature equation. Similarly, these large coccolithophores were offset by, about 3 or 4 per mil from equilibrium. And this is where the coccoliths just got left alone. Look at the size of these biological signals that are imprinting the chemistry in the liths. But as I've said, the advantage is we can grow these in the lab and we can start to modify the environment that they live in. And what was rather curious was that if we grew them under a variety of PCO2, in fact, I did these experiments because I was interested in their response to ocean acidification, but I thought, oh, well, we'll look at the isotopes as well and just see what happens. And what was curious is that these vital effects collapse as you go up to higher CO2 conditions in your culture experiment. So in the open circles here, I've got the large coccolithophores, and in the black circles, I've got the smaller, more lightly calcified coccolithophores. And so here's what we'd predict for equilibrium calcite, and you can see that these large coccolithophores are very much offset to light isotopic values at low CO2, but as you go up to the higher CO2 conditions, they're collapsing towards equilibrium. So we have actually the potential for a proxy here for CO2, actually in the offset between the isotopes in these two fractions that we can separate actually out from the sediments. And I was fortunate to have Harry McClelland as a graduate student recently. He's over in the States now. And he performed some more culture experiments and very much confirmed that we had this change in the vital effect with DIC or with CO2 of the cultures. And he was able to build a model here, which a very simple, essentially mass balance model of the cell, looking at the fluxes of carbon coming into the cell, being partitioned between the photosynthesizing chloroplast and the calcifying vesicle. And he was able to model this trend of the isotopes in terms of 3 main factors. So what's really driving the large guys to light isotopic values at low CO2 is this very heavy calcification there. You've got calcification, which takes out the heavy isotope and essentially leaves the reservoir light, and that becomes increasingly light as you take more calcium carbonate out of the solution. As you go to higher or less, so this is a very high utilization of that internal carbon pool, so where you're essentially driving Rayleigh fractionation a long way along the curve. We've then got this flux times the isotopic composition of leakage from the chloroplast, and that peaks here because this is where we've got the largest flux, which is isotopically distinct. We see this collapse down at either end because essentially here you've got very little flux. You're utilizing almost all that internal carbon pool. And here you've got a very high rate of income of carbon. And the third factor which goes into this model is looking at the isotopic composition of the carbon that's coming into the cell. So changing from predominantly a CO2 source here, which is the natural source of carbon for the cell, and going to the more isotopically heavy bicarbonate. So these 3 factors build in to give us these curves for the different isotopes in these different size fractions. So they depend on calcification and they depend on this degree of utilization. And this is where we think the CO2 comes into that signal. And the higher the CO2, the lesser the degree of utilization. And so you essentially flood the cell and see no isotopic variance depending on this physiology. So this was something that was very much taken up by Bolton and Stoll in 2013, where they were looking at the difference in these isotopic values between the small fraction, which is in the sort of blobs here, where they've normalized everything to the colored blobs, and then looking at the offset of the large coccolith fraction from these blobs. And what you see over the Cenozoic coming towards the modern day is this emergence of this vital effect in the last 10 million years. And they took this to show a decline of atmospheric CO2 at this time. Now, Harry would say, you can't say it's CO2, we can only say it's a change in utilization. We must be very careful. That utilization can be affected by growth rate and by the size of the cell. So it's very hard to make a direct translation to CO2, but certainly something is happening to the physiology, the physiological differences of these coccolithophores over the last 10 million years. And again, Clara Bolton has some really nice work looking at the size of the coccolithophores through time, the size, normalized thickness of the small coccolithophores through time, and is able to show that over this 10 million year period where we see the vital effects emerge, we also see a decline in the size of the calcite liths, indicative of decreased calcification. It's curious, in the larger coccolithophores, we don't see such a trend in the size. They seem to stay pretty much constant over the last 60 million years. And we think this is because they've got different, oh Lord, sugar. I've missed a green, there was meant to be a partially filled green circle here. So what we think is happening is that the smaller coccolithophores are adapting to declining CO2 by getting small. They're essentially getting smaller and smaller, increasing their surface area to volume ratio. And there's also good evidence that they've got a good CCM. So they're able to kind of keep their internal pool of carbon full. And this is why we don't see their vital effects changing, particularly over time. The coccolithaeals, by contrast, have stayed very large. And what this means is that they have evolved to this increased utilization of their internal pool. They haven't got a full internal carbon pool. They're sort of running on empty almost. And that's allowing this vital effect to emerge. We've done a little bit more work. This was with the fabulous postdoc Renee Lee, who was happy to sort of join me in trying to blend biology and geology, where we were actually able to pull out of the liths of these coccolithophores the molecule responsible for their calcification. So this is a long chain polysaccharide. And we were able to show that the biochemistry, essentially the uronic acid, so this is the carboxyl group of the polysaccharides, which is binding to the calcium and enabling the biomineralization. So this is really sort of biochemistry in the past. We can get these molecules out of these liths going back 100 million years. And we were able to show that in those large coccolithophores, where their internal pool has got smaller and smaller, they're not able to concentrate the carbon. They actually modify the biochemistry of this polysaccharide and change its charge to maintain their calcification when their carbon supply is limited. And we're able to show this evolving biochemistry over the last 100 million years. So this is really getting biochemistry out of ancient fossils and being able to show its functionality. So that's where we've got to with the coccolithophores, this real image of paleophysiology, I think. And I was recently asked to actually try and write a review of that evolution of these vital effects. So I sort of had to go back to my 4am history, and I was trying to think, well, what's the best way of trying to figure out if a 4am vital effect changes? Unlike the coccolithophores, which have to live in that photic zone, the 4ams have got many different modes of living, and they can bob up and down in the ocean. So how do you really say when they're bobbing up and down versus when they're really displaying a different signal? So I thought, okay, to look at evolution, I've got to look at the symbiont-bearing foraminifera. So the 4ams are amazing organisms. They're animals, essentially. They feed on prey. But they have, living off these pseudopodia around the shell, they have thousands of symbionts. So these can be dinoflagellates or chrysophytes, and those are photosynthesizing. And there seems to be this incredible interplay between the symbiont and the calcium carbonate of the shell. These are wonderful pictures taken by Howie Spiro and David Lee. Oh, Lord, what's happened here? Oh, yes, that's right. So, sorry. So looking at the planktonic, so I again looked for the planktonic foraminiferal delta C13. This seems to be preserved very well. This was a wonderful paper by Izard et al., where they compiled a whole bunch of data. I was very happy Harry had some delta C13 data that was contributing to this. And looking at 2 foraminifera, what we see is a real dependence of their vital effect on the size of the foraminifera. So tending to light isotopes at small size, and tending up towards, this is what we think the delta C13 of DIC is, and so equilibrium calcite is about 1 per mil heavier than that. So we can see the trend of these, the delta C13 values with size in these symbiont-bearing foraminifera. This was classically attributed to the fact that as you get bigger, you get more symbionts, and they make the calcite isotopically heavier. I'm going to revisit that as we move into the next couple of slides, because actually the unusual part of the signal is a tendency to light isotopes at the small size. So the question is, how do these vital effects evolve? Can we actually say something about foram biology through time? And this was data very kindly provided by Brett Tipple, looking at symbiont-bearing forams over the last 64 million years or so. And what was extraordinary to me, so these are looking at Morozovella and Acarinina forams that have been attributed to being symbiotic. And these are the more recent Globigerina and Globigerinoides, I think, were the two forms. But what was curious to me that actually there's scatter in these data sets. But if I look at the offsets between these two different forms of planktonic forams, which you think are symbiont bearing, this offset is actually pretty constant. It doesn't change at all through time. And this to me, I was kind of interested in this. I thought, surely we've got this incredible biotic association. There must be changes with carbonate availability or carbon availability. But really, this is very much constant through time. And again, I was lucky Nick Shackleton had done some fabulous work in the 80s where he recorded the isotopic values of a whole range of sizes going back in the Paleogene. And this is also work with Stephen DeHont and Jim Zakos. And what was curious to me was that the isotopic values with size match exactly the trend with the modern day size relationship with delta C13. So in the red and the blue, I've got Paleogene, what are inferred to be symbiotic foraminifera, and that's plotting on top of our modern day ocean compilation from the Atlantic. And really there's no change in this vital effect through time. And so what does that mean? What does the fact that there is no change in the vital effect mean? I think it means that this symbiotic relationship is extraordinarily successful. And what's also shown here, well, sorry, I have a couple of things to say. So one is that I think our understanding of how the symbionts behave is not built into the models that we have at the moment. And that actually we can account for this trend in terms of the amount of respired CO2 that's mixing with seawater to provide the calcium carbonate. And actually this trend is literally matched by the surface area to volume ratio of the shell. That's all that is in that relationship. So as you change your surface area to volume ratio, you change your proportion of respired CO2, which is contributing to the shell and making it light. And so actually this is a geometric relationship. It's as simple as that. So this is fabulous news for using forams to look into the past, because it really means we understand the vital effect and it does stay constant through time. What's also curious, though, is that the modern day forams get very much bigger compared to the Paleogene. So these are going up towards about 1,000 microns. And Nick Shackleton's largest size fractions were only about 400 microns or so. And this was something that was captured by Daniela Schmidt, where she looked at the average size of forams going over the last 60 million years or so. And although at the high latitudes, the forams stayed very much at about the same size in the green blobs here, at the low latitudes, the forams were actually getting a hell of a lot bigger. And that's very much seen in that modern data set. And so it really seems that this symbiotic relationship is allowing the forams to actually get bigger and to become more successful. Daniela attributes this to increased stratification of the oceans that gives more light and allows the symbionts to flourish. But that means the forams, and I think in some diversity curves, we see an increase in this foram, the number of foram species in the last 10 million years. So there's really something changing, allowing the forams to flourish. But what's curious is actually this is at exactly the same time as the coccolithophores are diminishing their calcification. So I'm sort of curious as to whether there's actually a tussle between these two mineralizers for the available alkalinity. What's also curious is Peterson and Bachman back in the 1990s showed this enormous increase in the mass accumulation rate of carbonate that goes alongside this increase in the foram size. So it could be this is actually changing the carbonate cycle in the ocean, and just very curiously looking at, I forgot to put the thing on here. This is Heiko Palike's fabulous compilation. But if we look at his measure of the CCD over the Cenozoic, then again we see in the last 10 million years the CCD actually rises, and this is the only time actually that it decouples from the Zakos curve. So through all of the rest of the Zakos curve, every time we see the oxygen isotopes get heavier, we see the CCD drop. Every time the oxygen isotopes get heavier, the CCD drops. And it's only in this last 10 million years that we see the CCD rise at the same time as the climate continues to cool. I think that's really curious and suggestive of a big change in the carbonate cycling in the last 10 million years. So I have to thank Mikhail Hermoso, Harry McClelland, Renee Lee, Despoina Mavrodou, and of course Harry and all these wonderful people for giving me some money. I've probably talked far too long, but I'll leave my conclusions up for you to read. Thank you very much.
We've got time for a couple of quick questions. Yes, one at the back. Could you just wait for the microphone and we'll capture you forever on film.
If we take Plantic 4M evolution a little bit further back into the late Cretaceous, do you think the rules will still hold if we go back into the late Cretaceous? Because there are very large Plantic 4Ms in the late Cretaceous when it's not like the Miocene.
So which rules do you mean? Do you mean the control on the vital effect?
Rules of the vital effect. I mean, it's really striking that certainly during the early Cenozoic and through into the Miocene that those Paleogene 4Ms are looking fairly small. I'm just wondering, do the rules get reset by the Cretaceous? The end of Cretaceous
I see
extinction?
what you're saying. Oh, crikey. Well, I think the question is, I was trying to figure out when the first symbiotic 4Ms arrived, and the challenge is that in the old literature, the symbiosis is inferred from the carbon isotopes. And I guess what's curious to me is that the carbon isotopes are, well, the question is, where's the respired CO2 coming from? Is it coming from symbiont respiration or is it coming from foraminiferal respiration? But the symbionts aren't doing to the chemistry of the shells what is classically written in the literature. They're not making them heavier than equilibrium. The impact of the symbionts is actually making the shells lighter. I think that somehow this there's the light input to the shell has to be accounting for that vital effect. So in terms of putting it into the Cretaceous, I don't know quite now how we infer which are the symbiont-bearing 4Ms and when they started. My sense is that you can look to the most positive carbon isotopes of the planktonic 4Ms because those are the ones that are shallowest in the water column if you've still got a biological pump. So my sense is that if you look for the heaviest carbon isotope 4M of the same size, you will find that what we might infer is the symbiotic 4M. But I just don't know quite when that symbiotic association started. But what's curious actually is that if you go through some of the ESARD compilation and plot all of the species, they kind of all fall onto this same trend.
Very good, thank you. I think we're going to.
Sorry, I talked.
No, don't worry.
Long,
no,
sorry.
It was great. So, Rod, thank you very much. That was. Okay, we now have Tim Elliott.